This is the presentation of the project that Biology 1360 class has been working on in spring 2021. I'm coining my project Mutation Identification and Classification of DDR Mutants in S. palmbi yeast. My name is Amanda Lugo and I am under the direction of Dr. Li Huang. This table is table 2 in the paper. It's titled Length in Millimeters of Wild Type AL Mutant and FK Mutant in S. palmbi cells. This graph shows the S. palmbi yeast cell type, which includes a wild type and 2 mutants, AL, which is mine, and FK. It also shows average cell type, standard deviation, and p-values of each of the categories. The data supports differences in length in millimeters between HU negative and HU positive S. palmbi samples in wild type, AL mutant, and FK mutant cells. In all cases, the HU positive cells were longer than the HU negative cells, with a p-value of 1.39 times 10 to the negative 11th for the wild type cells, 9.8 times 10 to the negative 9th for AL mutants, which is mine, and 0.002 for the FK mutants. Mainly, the main idea of this graph is that HU positive S. palmbi cells are longer than HU negative cells. In my paper, this is referred to as Table 3, which is the average cell length of S. palmbi wild type and mutant cells. So this data confirms that there's substantial variances in length in millimeters between the HU positive and HU negative. You can see that the yellow color is HU negative and the gray color is HU positive. And by the bar graphs, the HU positive one is much longer. Overall, the HU positive S. palmbi cells were much longer than the HU negative. All 3 p-values for each dataset are statistically significant, supporting their significance. Alrighty, so we are on to table 4, which is the fluorescence microscopy table of the wild type, AL mutant, and FK mutant. In the Y column, you're just going to see the yeast type, which we have the wild type HU positive, wild type HU negative, and same with the AL and the FK mutant. On the x-axis, we have DNA distribution of daughter cells and the relative GFP production of the cells, and also what mutant type we think that it is. So this is a comprehensive qualitative observations gathered from the S. palmbi mutant cells, which mostly attempt to condense the visual appearance of the mutant cells under the fluorescence microscopy. Using the fluorescence microscopy, cells can be categorized as either wild type, sensor transducer, DNA repair, or cell cycle mutants. Each mutant varies by 2 deviances, DNA distribution and the RAD22 GFP levels within the cell. In the wild type, we have equal distribution of DNA, and we have increased GFP within the HU positive. This is our baseline right here, and then in the AL mutant, we have equal distribution in HU negative, but unequal distribution in HU positive, and also we have more GFP in the HU positive. So these indicate that this mutant is a cell cycle arrest. In the mutant FK, we have equal distribution in the HU negative and unequal distribution of DNA in the HU positive, and also we did not have a significant increase in GFP and the RAD22. So we conclude that this is a sensor transducer mutant. Here we have the wild type S. palmbi yeast in the fluorescence microscope pictures. In these pictures, you can see that there's a mix of long and short cells in both HU and HU positive, with mostly long cells generally. They have a high RAD22 GFP presence in the nucleus, and they also have equal DNA distribution in both HU positive and HU negative. In this project, we used this sample and these pictures as a baseline to compare the other mutants off of. Here we have the FK mutant S. palmbi yeast in the fluorescence microscope pictures. In these pictures, you can see that there's generally short cells in both HU positive and HU negative, and they have a low level of Rad22 GFP presence in the nucleus, and they have equal distribution of DNA in HU negative but not HU positive. The mutant yeast FK is a sensor transducer mutant because cells are short, DNA is unequal, and the Rad22 GFP levels were low in both HU positive and HU negative. Sensor transducer proteins are the earliest phases of the DDR pathway and activate initially before DNA repair and cell cycle proteins occur. This is why there is low GFP. The average cell length for HU negative is 9.6 millimeters and 14.2 millimeters for HU positive. The p-value for these two groups is 0.002, which is smaller than 0.01 and indicates statistical significance. I think the p-value is slightly higher than the desired because the sample size was small. More length data could give us a better average. Rad22 GFP was low in both HU positive and HU negative of this same sample. Sensor transducer mutants cannot detect DNA damage, so low Rad22 GFP will be present in the fluorescence microscope pictures. Unequal DNA distribution is due to the inability to repair DNA in daughter cells. These mutants fail to activate DNA repair and pause the cell cycle at the G2 checkpoint so that the cells appear shorter. Here we have the AL mutant of the S. pombe yeast in the fluorescence microscope pictures. In these pictures, you can see that there is a mixture of short and long cells in both of the samples HU positive and HU negative. They have a high Rad22 GFP presence in the nucleus in the HU positive but not HU negative, and unequal DNA distribution in the HU positive sample. This picture is a zoomed in picture of the AL mutant HU positive sample to just show the Rad22 GFP levels. The mutant yeast AL is a cell cycle arrest mutant because GFP is high and DNA distribution is unequal in HU and cells are short. These mutants do not stop at the cell cycle at the G2 checkpoint and the DNA is not repaired, which activates the Rad22 protein and glow in the microscope. The average cell length for HU negative is 11.4 millimeters and HU positive is 24.2 millimeters. The p-value for these two groups is 9.83 times 10 to the negative ninth, which is lower than 0.01, which indicates statistical significance. DNA distribution in the daughter cells is unequal in the HU positive S. pombe AL mutant samples. The unequal DNA distribution is due to the lack of time to repair the DNA that would cause premature splitting of the cell and thus unequal DNA in daughter cells. Rad22 GFP was increased in HU positive as compared to HU negative of the same sample. In cell cycle arrest mutants, the cell continues mitosis but cannot stop the cell cycle to repair the DNA, so the cell is short with Rad22. The cells that have increased length in our sample are due to a mix of wild-type yeast cells in the sample. If you note the fluorescence microscope pictures above, with the Rad22 GFP are shorter cells compared to the long cells seen in the pictures which don't have any Rad22. This type of mutant detects the DNA damage, which means a higher Rad22 GFP level, but it cannot delay the cell cycle to allow time to repair, so it's a shorter cell with Rad22 and unequal DNA distribution. Alrighty, so conclusion, UV exposure can lead to damage in DNA, which can express itself in various ways. When cells try to repair the DNA, it happens at various times in the cell cycle division cycle, which can alter the human genome a little bit differently for everyone. The resulting cells will look slightly different from the parent cells under a microscope, and one can speculate the time during the cell cycle that happened during the mutation. Using yeast is beneficial for many reasons, but this specific process helped us understand the cell cycle and mutations within that cycle better. It's important to note that there are many ways DNA can be damaged and thus mutated, and sensor transducer mutations are less commonly seen due to their role and timing during the DDR process. Cancer cells and yeast cells are very similar in their modes of the DDR process, which is why we chose them for this project.